Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Dineen, a certified genetic counselor and your host. To celebrate the year coming to an end, we are doing our annual episode, Genetics Wrapped. We are reflecting back on the top genetics and genomics news stories, advancements, papers during 2025. And we're focusing and emphasizing from papers that were chosen for the annual Genomic Medicine Year in Review 2025 paper that is published by the American Journal of Human Genetics. So we're going to link to that in the show notes. So if you're wondering, what is this paper we're talking about? We're going to link to that. And then also every individual paper we talk about is also going to be linked. So lots of work for my team there. But joining me for this discussion are two leaders in genomics. And very timely, we recently attended the ASHG conference. So it's perfect to have the president of ASHG weigh in, Dr. Sarah Tishkoff. And fun fact, it is the third time we've welcomed on the current president of ASHG for this discussion. So Eric has really made that happen. So Dr. Tishkoff is the David and Lynn Silfen University Professor in Genetics and Biology at the University of Pennsylvania, holding appointments in the Perelman School of Medicine and the School of Arts and Sciences. She is also the director of the Penn Center for Global Genomics and Health Equity in the Perelman School of Medicine. So thank you, Sarah, for coming on.
Well, I am thrilled to be here.
And as I already mentioned, returning to the show, Dr. Eric Green, former director of the National Human Genome Research Institute at the U.S. National Institutes of Health. Dr. Green's career has involved leading major genomics research programs, throwing in Human Genome Project, a lot of different projects over the many, many decades and years. And he holds the title for the most guest appearances here on DNA Today, including every year, as I mentioned, on our Genetics Wrapped episode. But more impressively, he was awarded the 2025 ASHG Leadership Award and had a very inspiring talk and well-deserved standing ovation. So congrats, Eric.
Well, thank you. That was absolutely the highlight of 2025. There was a low light of 2025, since you did introduce me as the former director of the National Human Genome Research Institute. So we should just get out there right away. The idea that this is a new title for me. 2025 brought the end of me being the institute director when the new Trump administration started to make some pretty substantive changes at NIH. I was the first of now a half dozen or so institute directors who have been removed from their leadership positions. So I am now retired from federal service, but quite active in genomics and genomic medicine, and continue to do a lot of things that, including following all the exciting things happening in genomics.
Yes. And can I just say, it was so well deserved. And not only, a leader in the field, a mentor to so many future scientists who are going to accomplish so much, current and future, but also has been a major partner and friend to ASHG. So it's really been a true partnership that we've really benefited from and we really appreciate. So very, very well deserved.
And I really am proud of, in my 15 years as NHGRI director, I really did work hard, and with my ASHG leadership partners and the various presidents, to really elevate the positive relationship, major collaborations. There's just so many examples, and what we're going to talk about this year in review paper is just one of many examples of stronger collaborations between the leading funder of genetics and genomics research in the world and really the premier professional society associated with human genetics and genomics.
Yeah. And I would be remiss if I didn't bring up, because we are talking about the US government, of what is happening with the NIH impacts and grants not being there like they used to, research funding being pulled. And that was a big aspect of the conference this year. And Eric, you brought that up in your acceptance speech for your award that, Sarah, you actually introduced him. So it's very apropos that you're both here. Can we speak to that a little bit? Because it was bittersweet that, Eric, you received this very well-deserved massive award, but then most of your close colleagues couldn't be there in the audience, which was just really sad.
Yeah, I mean, let's just zoom out for a second, because I can't help but point out what a bizarre year we have endured. If you go back in time to the 2024 ASHG meeting, which took place in Denver. And at that time, just by calendarings lining up, the very first day, essentially, of the ASHG meeting was the election, which ended up with a result that many people found uncomfortable who were attending the ASHG meeting, which really cast a bit of a downer over across the entire 2024 ASHG meeting in Denver. And then if we reflect as we go back we go back to the recent ASHG meeting now at 2025 in Boston. First of all, everybody was reflecting on, wow, what a year it's been, and we're going to get to it, all the devastation, all of the policy changes, all the implications. But it also allowed us to sort of calibrate, what's gone on in a year. But then, one more just dagger in our heart, at least my heart, was the fact that the government was closed because of the impasse with respect to appropriations. And so that meant that there literally were supposed to be 75 members of the National Human Genome Research Institute, my ex-colleague, my former colleagues, my former employees. They were all supposed to be there. I got this leadership award, which was wonderful, but it was as much a reflection of this incredible team I've got to lead for so many years. They should have been there sharing that award with me. And not only were they not there, so that was sad for me, but I would also tell you that, and Sarah might know the exact number. I mean, there were a lot of holes in the program of
Yep.
speakers and posters, and they were supposed to have sessions with grantees to talk about all the complexities going on at it. So, once again, at the ASHG meeting, there was this cloud over, no federal employees or nobody from NIH was there, and so it was very bittersweet in that regard. But the world's changed a lot. Science has changed a lot in a year, and there's no question. A lot of conversations were going on at the ASHG meeting about that very fact.
And I just wanted to say, I think, I don't remember the exact number, but I think we had something like around 190 federal employees who were supposed to be there and couldn't. And I know that I don't, again, have the exact number, but there were many that were giving platform presentations or giving posters and couldn't be there. And the other thing is, actually, we were so excited because literally, I think it was about 2 weeks before the meeting, we were contacted by people at NHGRI. And they said, listen, we would love to be there and talk about some of the initiatives that are going on at NHGRI. So we set up the specials, tried to fit them in. I was like 7 or 8 in the morning on Thursday of that week. Everybody was so disappointed because we know how much people would appreciate that, but we're going to reschedule. So there is going to be a plan. We're going to reschedule that. It's going to be some type of a virtual event. Everybody can come and talk to people from NHGRI, and they can give us an overview of what's going to happen.
It's bewildering. It's head spinning. I spend a lot of, I mean, if you ask me, what do I spend a lot of my time doing? I spend a lot of my time talking to colleagues, either in person, by Zoom, by phone, who just cannot keep up with all of the crazy stuff going on at NIH, and it hasn't let up. I mean, it's still continuing. There's still lots of questions. And so yeah, there's a lot of outreach that's needed just to keep people informed. And some of it's really, complicated, nuanced stuff that you got to talk people through it to fully understand the implications.
It seems to me one of the hardest things is uncertainty, right? It is, we're going through a period of tremendous uncertainty. We don't know who the new director is going to be or the directors. I think they're doing a search for 11.
13 directors are currently vacant, 13 of 27.
13.
27 institute and centers at the NIH that make up the leadership table, and 13 of them are acting at the moment. And now they've just recently, the news of 2 weeks ago, is that they're going to do a bulk search for all of them. So all 13 are open right now.
With 2 weeks to apply.
Well, with 2 weeks to apply, which is causing a lot of consternation, because normally I can tell you, having been involved in more than 20 such searches over my years at NIH for the highest level leadership. Normally, these searches are open for 2 to 3 months because they go out and involve a lot of hunting, where you call people who never thought they would be interested in coming to NIH and you talk to them about it. There's no time for hunting. And so it calls into question what's going to happen with bulk, high throughput, rapid searches for probably the most important leadership positions controlling the biggest pot of research dollars in biomedicine in the whole world. That contributes to what Sarah refers to as profound uncertainty.
Profound uncertainty coming on top of a very challenging year. And so we've heard from members, we know how hard this year has been.
Yeah. And I think a silver lining to all this happening is that for the past two conferences, it has brought people closer together of kind of having this common experience of these difficulties. But even with some of the presentations, as you mentioned, Sarah, just being like, okay, this is just open on the schedule now because it was an NIH employee that was going to present. We had some people step in that was colleagues. I think of Wendy Ullman, who stepped in and presented for her friends. And she said, I'm not taking questions. I'm presenting this. Please direct your questions to her friend that works at the NIH. I don't know their name.
I think it was Chris Gunter,
Chris Gunter,
recall.
yes.
Yeah.
Yes. Wow, you have a good memory. And I mean, so there was a lot of camaraderie, I think, when it came to that of last year. A lot of people commiserating over who was elected. And then this year of like, we're missing a lot of our core people. So I think that was an aspect that was that silver lining, I would say. And Sarah, you gave a wonderful presidential address, which I was not feeling well when you actually gave it. So I watched it virtually last night, actually. So I was able to make it to the rest of the conference, had to, rest up that first day. And one of the things that I wanted to pull out from it is that you're the 14th woman to serve as president, which I was very surprised by. I thought that there would only be a handful. But this is the first time ever in the ASHG's history that the entire executive board will be comprised entirely of people that identify as women. And people were like going crazy in the audience for that stat.
They took me by surprise. And I want to point out, you're surprised that there were so many, but I want to point out that's 14 out of 77. Okay, so while it's better than having none or just a handful, it's still not, it was still not as many. And it's great that we have more women in leadership, but we still have work to do. We need better representation of our entire community, our entire society, right? So it is super exciting, but we still have work that we need to do. We want to see more representation. And I think one of the points I made is I really do think it matters because, I showed like a photo of me from, what was it, like 1993 or something like that at New Orleans ASHG. And, looking back at that person then, would I have dreamed of being president? No way. And if you don't have role models, it's really hard to imagine yourself in that position, right? And so it is really important and it brings more diverse perspectives. It makes us a better organization. So it was super exciting. We still have more to do, but I am happy about that. And that was great how the audience responded.
Yeah, no, it really was. And I think just looking at different types of diversity when it comes to who the president is, one thing that I was learning about, because we're just meeting on this recording, Sarah, that there's a lot of different backgrounds in terms of where the president has come from. For those that are not as familiar with your research and your work, what kind of background are you coming from that you brought this year as the president?
Sure. Well, I have an interdisciplinary background in anthropology, human evolutionary biology, human genetics. And so I guess you could say human population genetics more broadly. And it is true that, a lot of the presidents have been more clinical oriented or medical geneticists, but we have had several people, Charles Rotimi, Neil Reish, Mary Claire King just come to mind, who are more in the population realm. And it is really important that we have a diversity of backgrounds also in terms of research focus be represented as well.
We emphasize it's something that I've always had great respect with the society about. Maybe part of it is I joined the society very early on as a genomicist because I don't really have any true credentials as a true human geneticist. I've picked it up as a hobby, but as a genomicist. But that was at a time where the society where genomics was just forming and the society. But I've just watched over the years around the board table, around the presidents, just the people that it has become, and particularly in recent years, a very welcoming, highly interdisciplinary society, and they reflect that in their leadership and their board. And I think that's a really valuable thing. Maybe because I grew up in genomics, which is so highly interdisciplinary and so highly appreciative of different types of expertise, recognize we need a big tent to make progress. But I think the society has really evolved in that same direction.
Yeah, I think as you say about we're trying to make it a Big Ten. So I mean, we have genetic counselors, we have people who are just purely educators, we have clinicians, we have basic science researchers, we have people studying evolution, we have people studying, clinical disorders and medical genetics. And so we really try. We're very purposeful about having this more inclusive, I would say, tent.
Can I ask Sarah a question? Because Sarah, both in her address that she gave, which was outstanding, and the presidential symposium which she put together, which of course was similarly outstanding. It really did, both of those obviously reflected her areas of interest, which have multiple dimensions, but at the core of it, or the common theme, is diversity and the importance of diversity and include, et cetera, et cetera. There was a certain irony that here you were being showcased as the president of society the very year that all of this was horrifically under attack by the U.S. federal government. And I'm just curious, Sarah, if that made you even more excited and motivated to showcase it, or did you feel like, did it give you any sort of pause about, how you were representing it, since you were also not just representing yourself, you're representing the society. So how did you approach that as you put all that together as
Oh
the year
boy,
went along? And I know that's probably a hard question.
that is a hard question, Eric. I would say very carefully. Because the fact is, as a president of a society like American Society of Human Genetics, I have a fiduciary responsibility. Okay, so that means I can't just talk on behalf of myself. I'm also talking on behalf of the organization. We get NIH funding, right, for our programs. We also have to be aware and cautious of, not losing that funding. At the same time, I think all of us at ASHG were just very proud that we were able to stick to our core values. So that was something that we could proclaim and that we're proud about, that we want, I forget exactly, but human genetics that serves all people essentially, or that benefits everyone. But I did have to be careful, but at the same time I couldn't ignore it, Eric. And I felt like if I totally ignored it, one, I'm not being true to myself, because my entire career, just to give you a little bit more background, my entire career has been focused on studying African genomics. Back at a time starting when very few people were doing that. They continue to be greatly underrepresented, but they were even more so when I was doing my PhD work. I did a short postdoc in Johannesburg, so I was in Johannesburg for a while. Have integrated field work in Africa with many African partners and collaborators, and then we do computational analyses and wet lab work. But having made that my career and talking about the importance of diversity, I felt like that had to be highlighted. So part of my talk was also just talking a little bit about my own research and how much variation there is in Africa, but also pointing out that these multi-ancestry studies are important, not just for health disparities. That's very important. That is a valid reason to be studying, is to help eliminate health disparities. But these studies help everyone. And so, a very important point I was trying to make is that, for example, it's been well documented when people do these multi ancestry studies, for example, looking at polygenic risk scores for trying to predict risk for disease, they do better. When they try to find map, when they do GWAS and they're trying to find map functional variants, they do better. I gave the example of, make sure I get it right, PCSK9, I think, is that right? Helen Hobbs did this beautiful work, and that was in like the early 2000s, I think. And what was interesting is she was studying these variants that knock out the function of this gene, knock out the protein, are most common in people of African ancestry. And it was by studying people of African ancestry that she found this. And they also happen to lower LDL. People who have these knockouts, natural knockouts, have lower LDL. That led to the development of a therapeutic, and that therapeutic is effective in everyone. So I really want people, I think people, and I'm hoping that people at NIH, I think most people at NIH, but at the highest levels now understand that having diverse cohorts isn't just a DEI issue at all. It really is important so that everybody benefits, number one, not just, people of one ancestry or certain ancestry. It helps all of us. And I really think that point is super important for
Well, I can
people to
tell you,
understand.
Sarah, that I lasted about 2 months into the new administration before my date for dismissal happened. And in those 2 months, some of the new administration people were seeping their way into NIH leadership positions. And I tried educating and tried explaining those things, especially from a genomics perspective, that, diverse genomic data isn't a DEI issue. It's a
Exactly.
medical imperative. That was one of
Exactly.
and I made
Exactly.
these arguments.
Right.
I don't think I made miles of progress. I made probably maybe millimeters or inches, but we
Well,
can't let up
yeah.
because there's social arguments, there's scientific arguments, and there's medical arguments. There's overlapping, but the people that there's a lot of misguided attitudes that need to be combated, and we have to be
Right.
really careful how we do it. And I know you're
Hmm.
the head of a professional society. I know it gets complicated. I can remember when Nancy Cox, for example, was president of ASHG during the Trump 1 administration, when there was a lot of pushback by the administration on immigration, once again, and the society took a very strong stand about the importance of individuals from other countries coming in and training in the United States for our scientific enterprise. But her name became associated with that, and that required that she faced some challenges as a result of that. So, these are tough things, is what I can tell you.
Yeah. But you did mention, there was also a mention, I think someone said about international, right? But I mean, that was the other absolutely devastating thing that happened this past year is international collaborations were cut. No advance notice, nothing. I mean, entire research programs just pulled out from under people. And again, just has a devastating impact because I think I would like to know, or we have to educate people generally broadly that international collaboration and training benefits all of us. It benefits us tremendously in the US. I wouldn't have been able to do my own research without collaboration and many other people. And it is so critical to us being leaders, the US being leaders in science. It really benefits everybody. And I really hope we can get that point across. And we're trying, because the other thing that we do that I've done a lot this year, which I had never done before, was to advocate for biomedical research and for NIH funding. I went to the Capitol a few times. I'm sure, Eric, you're used to this, but for me, I was like, when I first went there, I was like a little kid. I was riding that train that goes between, the Senate and the House. And I was just like so excited and a little bit intimidated, but it felt good to at least be trying to do something. And I think we just have to we have to continue, because one thing we heard from everybody, didn't matter if they were Republicans, they were Democrats, they said it's making an impact when people are calling and writing. And they're like, we want to hear your stories. And ASHG, I am so proud of the work that they have done this year. They have been so phenomenal in terms of, doing advocacy work and helping our members, giving them the tools so that they can be advocates themselves. And yeah, I think that's something I'm particularly proud of, actually.
And I mean, again, there's a lot of wet blanket topics we could talk about during this podcast from the last year. But on the positive side, Sarah's efforts and other advocacy efforts for NIH funding, in particular, has been effective. So as we get near the end of the year, we're now in another, I don't want to get too wonky, government wonky, but we're in a continuing resolution still. We'll see. And NIH still doesn't have its year-long budget, and hopefully it'll get one in January. Coming out of both appropriations committee from the
Yeah.
House and the Senate, it's basically a flat NIH budget when the president was calling for a 40% reduction.
Yes,
So
exactly.
there's a lot of problems with NIH right now, and some of it relates to visas and immigration and foreign funding and foreign collaborations, and there's lots of things to be and indirect costs. But the simple thing of just the sheer dollars, Sarah and all of us, and I say the whole community who have been working hard to educate both chambers of Congress, have convinced them that NIH is vital, and the thing they could do is give money. All the other things are things that are now, being fought between the executive branch, the legislative branch, the judicial branch, blah, 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 blah. Bipartisan support for a healthy NIH in terms of funding. And I think we could expect no matter what happens over the next year, funding won't be the problem. There'll be other problems.
And that's so key. Yeah.
And I want to I would be remiss if I didn't acknowledge, first of all, Karina Miller, who's a staff member at ASHG, Associate Director of Advocacy and Public Affairs, and Tony Winchell-Boris, who's the chair of the Government Public Advocacy Committee and the well-deserved recipient of this year's Advocacy Award. They worked tirelessly. And also, even things like going to Congress and educating them about what is genomics. What is it? Why is it important? What are some of the diseases that are being, addressed and cured, and the amazing work that's being done? Really, this year was about advocacy, advocacy, advocacy. And as Erica was saying, ultimately it was largely successful. There's still work to do, as we know. There's still work to be done, but that the work paid off.
Yeah, literally,
Absolutely.
it paid off.
Yeah,
Like
we
the
hope.
money,
We
the
hope.
money is there.
We don't want to jinx it, right?
Right.
They don't have a budget yet, but it's looking more encouraging at least.
Yeah, I think that's good headed into 2026. But before we get to 2026, I think it would be awesome to cover some of these top 10 genomics papers. Eric, usually I toss it over to you to give a quick elevator pitch history of like, what is this paper and how are the papers chosen from the last year? Like, what are the, maybe not criteria, but the factors and what's really looked for to be like, these are the top 10 papers?
There's a history associated with this. And by the way, this is the first year, the paper that's coming out in 2025, this is the first year that I'm actually not even a co-author. I wasn't
I
involved in it
noticed
because
that.
I had already left the government, but I've been involved in all the other ones. To really understand the context of something I'm very proud of, I'll take you back to the year 2011. 2011 was the year that the second of three NHGRI strategic visions, all of which have been published in Nature since the Human Genome Project came out. So there's one in 2003, the day the Human Genome Project ended, 2011, which I'm about to tell you about, and then 2020, which is the one we're operating on now. In 2011, it reflected Francis Collins had started the strategic planning process, and then he left, and then I took over the institute in 2009, wrapped it up. So the 2011 paper was really my first one as the NHGRI director, and this is where it's relevant to the year in review, is that it was the first strategic vision out of NHGRI that explicitly in the title talked about how genomic medicine was our guiding light, if you will. Or, that was the ultimate destination we needed to be heading towards. And it was really important to appreciate, like many times in the history of genomics, people didn't always agree. And I can tell you back in 2011, some people were a little critical of me as the new director, that I was pushing genomic medicine too hard, that it was too soon. And but I had very strong support of my advisory council, the highest level advisory group. And so when the 2011 paper first came out, it bubbled up from our advisory council members that, maybe we should set up a working group of this advisory council to really help define what were going to be the priority areas to really invest money in genomic medicine. And we really liked that idea. We had nascent ideas, but we liked the idea, as we always do at NHGRI and genomics, of engaging our brilliant community because they're always helpful. So we created something called the Genetic Medicine Working Group of the NHGRI Advisory Council. That working group has existed. It meets monthly. It has met monthly since 2011. Membership has churned to a large extent, but it's done a variety of things. And one of the things it started to do was, back in 2011, there wasn't much literature in genomic medicine, but that hasn't started to come out that we should we could be the source of curation. We could set up a website. So we just started curating and we would talk about it at every call. And we would basically say, is this paper worth featuring? Because you don't want to just throw every genomic paper. I mean, that you just do a PubMed search for that. So we would curate it and basically say the things that we thought was worth people looking at. And so we would do that. And I forgot how many years in, we'd have to look it up. But some number of years we said, you know what, maybe we should do a year in review, or maybe that may have been that American Journal of Human Genetics editor approached us. I forgot that part of the history. But then they just sort of got organized that once a year in December, they would publish a year in review such that, a few months earlier, the working group was getting ready for it. They would just look at what they've been curating and then through a voting scheme, pick the top 10, and then each member would write one paragraph summary, and then the whole thing would get stitched up and edited. And so I was involved in a bunch of these. I wasn't involved in this one, but is it a perfect process? No, don't take overly literally that these are the only 10 papers worth reading. It is just it's just an honorific. This is a working group that geeks out all things genomic medicine once a month, looks at papers all the time. And at the end of the year, it just votes on what are the 10 papers that rock their boat the most. And that's what it is. And it's fun, but again, people shouldn't take it too seriously that if you're not on the top 10 or you are, it's just a highlight reel, like any highlight.
And it makes my job much easier of coming up with what we're talking about on this episode because it comes to me on a silver platter from Eric's email. So that's really, really helpful. And there's a lot of themes that are coming out of this. I think the biggest one the focus is gene therapy. And so that, I mean, I have to start with my personal pick of the year, which is baby KJ, who had a personalized CRISPR therapy that seems to be working. So, Sarah, you had mentioned this paper in your presidential address, actually. So, why did you end up highlighting this one?
Oh my gosh, it was just so exciting because it was the first, or at least one of the first examples of truly personalized CRISPR-based editing. And in fact, I'm just trying to look again want to make sure I get the names of both the authors correct. So that is Rebecca Ahrens Nicholas, who is a pediatric geneticist at CHOP, and Kiran Musunuru. I forget actually if he has an MD or not, but he's been leading developing gene therapy approaches. He's at Penn, and also is going to be our incoming editor at the American Journal of Human Genetics. And they just gave a talk to our department in genetics. I just heard about this. I just literally was just astounded because think about what they had to do. So this baby comes in, very severe disorder, right? So they've got this, I think it's a urea related disorder. Let me see, it's called severe carbamoyl phosphate synthetase 1 deficiency, but it's deadly. Okay, without treatment, deadly. And children, my understanding is children who have this or are born with this, they have to have a liver transplant within year one. And even that could be too late and you're on a list and it could take a long time. What was amazing is they had to develop this personalized treatment that is based on the specific mutations that this baby has. So you have to design the CRISPR, guides and everything. You're testing it in cell lines. You're testing it in mice. You're ultimately testing it in a primate model for safety. You have to get it approved through the FDA. You have to go through all the very detailed IRB review and so on. And they were able to do that within about 6 months so that baby got the first treatment by 7 months.
Which is remarkable.
That's remarkable. Remarkable. And then nobody's done this, right? So you're kind of, they started with a low dose because they're being very, very cautious. And then they saw, okay, that low dose looks like it's effective. There's no severe negative effects. Okay, we're going to go to another dose. I think it was roughly 6 weeks later or something like that.
Yeah, it was when baby KJ was 8 months. So yeah, right around there.
Right. And I just do want to mention there is a paper they just recently published, the two of them, in American Journal of Human Genetics. It's more of a perspective. And what they're talking about is that it is very important to be able to have clinical trials. And so to have clinical trials, you kind of need more than an N of one. And so,
which is what this is. They basically personalized it for this baby and it went well, but we need to see it in more than one person.
Right. And so what they're proposing is sort of take an umbrella, use it they're calling it an umbrella approach, where there are a number of different disorders. I think they're saying PKU is one of them, for example, due to different mutations at this PAH gene. And they could be treated in a very similar way. So certain parts of what they call this gene therapy, or they actually, I think, called it drug treatment, gene therapy drug treatment, parts of that can be used for these other types of disorders. So if they can make them into an umbrella of similar ultra rare disorders and then get permission for doing these clinical trials, it could take us really far. And I think they either already received that permission or they're very, very close to being able to do that.
So would that be sort of like creating a pipeline so that you're like, this is how we do it for insert whatever disorder as long as it's one variant?
Well, not a ton. I think
that would be too much. That's too broad
right now, I think right now we're talking about ones where, first of all, it's mainly problems with the liver. And this particular approach that they are using is where the liver cells are really targeted, right? But the type of vector they're using, the type of approaches can be similar across these different diseases. Sometimes you just have to change this mRNA that they're using, it's 20 nucleotides long or something. You just have to change it, tweak it a little bit, but everything could have slightly different effects. And they had to be so careful about off-target effects. You have to know what else might this CRISPR editing be changing. So, and what I also was amazed when they came to talk about it were the ethics involved and how careful you have to be from the beginning about explaining this to the parents and what are the risks, which could be tremendous, and what are the benefits. And it was amazing that the parents were so enthusiastic about this as well. Yeah, it's one of the most exciting things because the other thing that I hope in my talk, I did talk about treatment for sickle cell. I mean, we're talking millions of dollars for every single time this gene therapy approach is applied. And if you're looking at the people who are being affected by this, people of African ancestry in the US, and I forget the numbers, but I think it's hundreds of thousands, if not millions of people in Africa, and they can't afford that treatment. So what excites me also is the possibility of what they call bespoke, gene therapy. Can we now get the cost down and get this to a point where it can be applied in places like Africa routinely? That would be like super exciting. But maybe that's for the upcoming part 2026 and later.
Yeah, maybe. We had Victoria Gray on episode 288 and 289, who was the first person that it was asymptomatic from sickle cell through the clinical trial that later that drug was, or that the gene therapy was FDA approved. And so it's remarkable. Go ahead, Eric.
No, I was going to, sometimes when we do these year-end things, I look for themes. I figure people can read the details by reading the paper. I tend to look for themes, and I also always like to throw in a historic context. One of the things I think is very interesting about, first of all, if you would have told me, 10 years ago or 8 years ago, 5 years ago, that we were going to have so many therapeutic highlights in our year in review in gene therapy, I mean, I would have said, you're kidding me. So, pause for a minute and just how remarkable it is that we're talking about this. But there's the theme I really want to stress, which actually relates to two other papers in the year in review, is this notion of we're just at the floor. We're not getting anywhere near the ceiling. Now, what do I mean by that? What we were just talking about, and what Sarah was explaining, the possible general toolkit development for these therapies, is that however excited we get about these exemplar early examples, we know the technologies are going to get better. There'll be more general purpose. This is the floor. It's only going to get better. Now, if I go back in time, now we went from the therapeutic side. Now let's go to the diagnostic side. When we started doing, just whole genome sequencing for routine diagnosis of rare genetic diseases, we were so excited when we found one and then two. And then we were so excited when it was 20%, 30%, 40%. Maybe now it's getting up to 50%. And I constantly tell people when I give talks that this is not the ceiling. We'll figure out the other 50%. Some of it's going to be because of biological complexities, but some of it's going to be because we're going to get better at this with respect to our tools. And what are two of the papers, which was highlighted a lot this year, long reads, that if you do all these diagnostic things that are quick and cheap with short reads, well, yeah, you find a lot of stuff. You skim a lot of cream, but some patients you can't figure out. Well, guess what? You throw in long reads and it gives you a window into genetic changes that could lead to rare genetic disease you missed by short reads. And
so what is the difference, Eric, between the short reads and the long reads for people that have heard this concept but don't understand it?
Short reads are going to be really, for example, short reads will be really good if the mutation itself is like a single nucleotide change or a small region of DNA. But if you're dealing with structural changes or any sort of longer range changes, for a diagnostic less than $1,000 genome sequence, you don't get all that. But long reads get you long range information, which in many cases reveals long range mutations that only detected by long read that could cause a rare genetic disease. So what that means is now we've got to get it cheap enough, but all that'll happen. But the papers here demonstrated that now we're starting to push our 40, 50% up to 50, 60%, and we'll just keep going. Technically, it'll get better. And then I think the way of coming, the other angle will be that we'll better understand the functional landscape of the human genome, because inevitably there's probably the answer sitting there. We just don't know how to interpret the answer yet. But, we're working hard to interpret the human genome every day. So I don't know, again, in both ends of the spectrum, diagnostics, therapeutics, everything we see, we should be excited about, knowing it will only get better every single day.
And just adding on to that, another thing it helps with is you'll get structural variants, which are not easy to get with the short read data. It can help you get through difficult to sequence regions, right? So there can be like very repetitive, and you might miss, it might be the causal mutation there, and you're going to miss it because without that sort of technology, you can get phase haplotypes, right? That can sometimes be really helpful. You get methylation from some of these technologies, and I think there was a paper, I think it was in American Journal of Human or no, it was in ASHG Advances, where they looked at using the long reads to characterize CpG differentially methylated sites and how that could be useful for identifying disease risk. So I agree. I mean, that's a super exciting area.
And that's a topic that was really brought up a lot at a conference Eric and I were at, AGBT Precision Health, which Eric, you were on the the recap of that, one of the segments, and that it was just talked about so much. So that's something else that I was like, even going into reading this year in review paper, I was like, I had kind of already had that as a thought of what to bring up during this episode, because I just kept hearing about long range genomic sequencing, long read genomic sequencing, and how we're headed towards that. And one of the papers is proposing like this should be first tier sequencing at some point. But right now, are we more looking at this as like a second tier, like get your short read genome sequence? If you can't find it, then do we do long read? Is that where you think we're at right now, Eric?
I think we're at a metastable place right now. It's gonna costs are gonna drive everything. I mean, and so when and by the way, companies are working on combined technology. I mean, they're coming up with ways. All these tool companies recognize the need for both, and either that's going to have to come from two companies or one integrated, all this. But it's just one of these things that will struggle in the short term because it'll all relate to who's paying and how much it costs, blah blah blah. But in the technology is going to drive us forward, and I think we within 2 to 3 years are going to see a different set of options. Now, I don't think and by the way, let's keep in mind that the incredibly brilliant community of genomicists falling under the telomere to telomere consortium or the telomere to telomere, workforce, they are, I know many of them well, and they are working away, you know, doing miracles. But among the miracles they're working on is how to bring, what they've learned how to do, which to create a reference sequence, which is expensive by comparison. But what could they do computationally and with the right technologies to be able to get us closer and closer to a telomere to telomere diagnostic genome? And that may sound science fiction, but you know what? Everything we're talking about today sounded like science fiction 5 years ago. I just want to keep quoting.
Yes. Yeah, for those that haven't watched Gattaca, I watch it every couple years, and every time I watch it, science fiction movie with Ethan Hawke and Uma Thurman, and every year there's an aspect to it that is now real that wasn't before. The movie came out in 1997. We have a great episode to link to in the show notes where me and some classmates when we were in grad school just like talked about the movie and what's accurate and what's not. But it really is true. I think as we go, like, especially of both of you being in the field longer than me, of just how much you've seen that you're like, I did not think I'd see this until end of my career retirement, and like it's already happening.
Do you want? It's funny you should say that. Can I veer a little bit, or I can wait until later?
Please. No,
Okay.
do it.
So I was just going to say, following up on the long read sequencing, that was one of another paper I pulled up from this past year that I thought was great. It was published in Nature. Glenys Lugsden was the first author, talking about complex genetic variation in nearly complete human genomes, and she's part of that consortium that Eric has been talking about, where they're generating more complete reference genomes from diverse populations. That's super, super important. We need to increase the representation from different people of different ancestries globally. Because the other thing is, once you have that, it's also much more informative when the rest of us who have this short read data, we can map it back to that. And so we can learn a lot just by having those reference genomes. Now, you mentioned Gattaca, and I was like, okay, I was reading this last night. I'm just trying to see where I wrote this down. I come across this paper, okay, by, I don't know, Eric, if you've seen this. It was published in the last year by Peter Vischer and colleagues in Nature, and it was called Heritable Polygenic Editing, the Next Frontier in Genomic Medicine. And they mention, like, Gattaca came up.
In the paper.
I believe it was in that
My
paper
God.
that I saw Gattaca mention, because what they were talking about, which frankly terrifies me, but I appreciate that they at least are even talking about it, and they tried to do some mathematical modeling and simulation. And the whole point was, they're saying, we believe that in the next, I don't know, like 10 to 20, 30 years, people could potentially be doing modifying embryos for polygenic traits. Okay, so heart disease, schizophrenia, diabetes, and so on. So they do kind of almost like an exercise. Like, how informative would this be if we were to change, let's say 10, we're going to say 10 of them, depending on the different type of complexity of the disease and the effect. it's a little scary when we start thinking, and then they go into the ethics and so on. But I mean, we have to at least talk about it because this could become a reality. I mean, so that's
Yeah.
part of our future. Like, I don't think that's even going to be 2026, but it is going to get to a point where there could be potentially editing for polygenic traits, not just Mendelian
Which step
disorders.
one of that is sort of happening. There is a company called Orchid, and there might be others as well. That's the one I'm familiar with, that does polygenic risk score analysis on embryos so that couples can prioritize which embryos they're implanting. From what my understanding is, they're not doing any editing, but they're just saying, here's the information, which embryos do you want to prioritize? But that's like a preview of what you're talking about, of actually then saying, well, these embryos have this chance of having a cardiac event like we see in Gattaca, or, just
And now
different
edit it.
factors.
And then,
now
I
edit
mean,
it
that's
to
a whole nother
optimize.
level. That is a
Yeah.
whole nother
By
level.
the way, a variant form of this, which I just learned about at a meeting I was at, the Mexican Society of Human Genetics meeting. And I think there's things maybe happening in Mexico that aren't allowed to happen here in the US. But if you have a set of embryos and you're prioritizing, even before we get to editing, just prioritizing based on polygenic risk scores, what you are prioritizing on can give you pause in terms of how strong the science is. And one of the clinical geneticists, or a couple of them I was talking to, was saying that some of these companies are advertising the ability to prioritize the choice of embryos based on intelligence. I mean, it's one thing to
That's
do this based
scary.
on a medical condition, but to do it based on intelligence. Now, again, we're not modifying, but what I worry about when I hear stories like that, I worry about, the misperceptions around what is not scientifically valid. And the last thing we need is to have overhype with genetics. And it just really, that story gave me chills.
And
Yeah.
not to mention the fact we do not have, I'm sure Eric will agree with this, we don't know the functional variants. We don't know, like majority of cases, we don't know the causal variant. So until you figure that out, and that's where there have been many advances in 2025 with people doing functional genomics and identifying what are the causal variants, because that is going to have to be the first step before we even get there. And then don't forget about pleiotropy and the fact that something that you think is so-called bad might have a positive effect. And I, as an evolutionary geneticist, I could tell you some things become common that may have a negative effect because they might also have a positive effect, or they might have it in a certain environment. So, like I said, we're a long ways away, but I think trying to identify functionally important causal variants is very important.
Yeah. And just knowing like how much I'm so curious about, especially polygenic risk scores for embryos, of like how confident are we with these polygenic risk scores? How much research has gone into each variant and the weight that each variant has that goes towards these disorders? Maybe we'll have to explore that on a whole episode because I'm so curious. But within this embryo space, one of the papers in the year in review was looking at pre-implantation genetic testing, reducing the risk of mitochondrial DNA diseases. So I was surprised to see this because this is kind of, I would say more of an offshoot of like another aspect to PGT. Did either of you look at this paper?
I didn't look at it in detail. I mean, it didn't surprise me when it popped up. I think a lot of these, the mitochondria, which doesn't have a big genome, so never caught a lot of my attention.
Right.
But a lot of these things come as a byproduct, the ability to do this. And there's a lot of genetic diseases, Kira, you know better than me, that emanate from mitochondrial mutation. So the idea of being able to do selection, embryo selection based on that makes a heck of a lot of sense. And I also think we should, a lot of efforts, we were talking about some of these efforts around gene therapy related to the nuclear genome, but there's a lot of interest and a lot of work going on to try to understand therapeutic options for mitochondrial disorders.
And it brings up that concept of like the 3-parent baby,
Exactly.
of like having a donor embryo, but then not having the nuclear DNA come from
Correct.
that egg and just having the mitochondrial. So yeah, I think that's a very kind of interesting area, but really complex because it's not like you can just biopsy, oh, this is what one mitochondrial has. There's so much different, I want to say viral load, but that's not the right concept.
Still, the number of the mitochondria per cell are just tons, and so no, it's
Yeah,
not
there's
a
hundreds. It's not
And
like when
heteroplasmy, that they can have
heteroplasmy, that's
different,
the
yeah.
term. Yeah, and like when you think about in textbooks, it's like you can only capture so much, but usually there's 2 mitochondria. So for many years when I was in high school, even going into college, I was like, oh, there's 2. And then at one point it was like, there are hundreds in a cell. And I was like, what? So yeah, that much more complex than it sounds, even though there's only 30, 40 something genes there. I'll fact check in the show notes on that. But I think with the polygenic risk score, to keep going with that for a moment, I was interested by the study looking at this, not in the preconception embryo space, but in just reducing cardiovascular events in adults and kind of the long-term study there. Because when I think of polygenic risk scores 10 years ago, it was kind of like, oh, this is a cool concept that someday we'll be able to use. And I think that someday happened like a couple years ago.
Well, and I think so. What caught my attention this year, and I talk about polygenic risk scores in all my talks, but and in my last few years as NHGRI director, we implemented a number of major studies to look at polygenic risk scores and generalizing to different ancestry groups, et cetera, et cetera. I always would categorize it as it's a research and development. It's an under construction area. You can show a little construction sign. I don't think it's ready for clinical implementation. I kept saying it. I thought it was interesting that earlier this fall, I think it was even the fall, the Broad Institute's clinical lab they now sell a test. It's for cardiovascular disease. I mean, they sell a polygenic risk score test. And, that's kind of moral equivalent of good housekeeping. Now, this isn't a fly-by-night operation. It's not a direct-to-consumer. This is a legitimate clinical lab. This is the Broad Institute. They don't use their name lightly. And they're providing access to this clinical test for, I think it's like 8 cardiovascular conditions or something. So that caught my attention, and I'll be very curious to watch the literature. It's not surprising to me. I always would say it's not a matter of if or when. I think polygenic risk scores will be bounded. I think Sarah's point's a really good one. We got to be a little careful getting too far over our skis without knowing the information about the functional variants. But, if it's a matter of guiding the clinical management of somebody and you just to work in your favor, what the heck? So I think we might be seeing a transition.
Although what worries me is that it could exacerbate health inequalities and disparities because and one of the things I would say also in the past year, there were a lot of PRS related studies that have shown that when done, these are calculated from giant GWAS, typically in people of European ancestry. And then you try to apply it to people from other ancestries and it just doesn't work well. So that test, I'd really love to know how well does that work in, people of diverse ancestries versus Europeans, because you could be giving wrong information. And even if it's wrong information in the way of that, it's not very informative, or it's like, oh, or maybe they interpret it as, oh, you don't have high risk, but maybe they really are at high risk, but it's just not tailored. So I do think that is an area where there've been a lot of papers, a lot of studies investigating, one, how transportable they are, but coming up with better methods that will be more effective across people of diverse ancestries.
Completely agree.
Yeah, I think that's where your research, Sarah, is so important, and that we can't sleep on this. I don't think we actually mentioned it verbatim, but the Pan Genome Project, that's really important in some of this work that Eric taught me about on a previous episode. And I think that, yeah, we don't want to exacerbate what's already happening. So we need to be even more on top of being inclusive with databases. And, we have this problem of, this is how our field started, is looking at European genomes. And it's like, we've really got to be able to incorporate more to be able to provide care that is more equal. One of the other papers that I actually had seen just in my LinkedIn feed of people talking about a lot the last few weeks is doing pharmacogenomic testing specifically for a variant or a gene. I'll check DYPD. Did you guys come across this?
I saw it go by. It wasn't one of the ones I focused on as much, but
yeah, it's looking at clinical utility of pharmacogenomics in chemotherapy for GI cancers. So basically, when someone's diagnosed with one of these GI cancers, they should have this pharmacogenomic genetic testing done to make sure they don't have one of the variants. That is counterintuitive to then prescribe because there can be adverse reactions. So I'm hoping that this is the first of many. And we did a really good episode with Josiah Allen. We'll link to that in the show notes, that really talked about how this is important. And there's a push for this area to be recognized more within the pharmacy not just industry, but within people that are pharmacists and having that be a recognized specialty. So I think that hopefully this is, I don't want to say the first, one of the first of many where there's going to be a checklist of before you get this drug and it's prescribed to you, we're making sure you don't have this specific genotype that's going to have a higher risk for an adverse reaction.
The truth is, the cancer arena is doing better around pharmacogenomics in many ways than the other areas of medicine. I mean,
Agreed.
we're still working on it.
Including like psychiatric too, which is surprising.
Yes.
I'm sure I've said like in an episode years ago that I thought that the first area to really widely adopt it would be psychiatry.
I thought that too.
So I just want a couple of things I want to follow up here. One is that, again, I'm going to focus on disparities because I really think there's a major bias towards these pharmacogenomic studies being done in people of predominantly European ancestry. And we are missing it. And one of the reasons I know this, we just started looking at, we have a very large whole genome sequencing data set from Africa. And we were interested in pharmacogenomics. So we started looking at, do we find predicted deleterious or strong effect variants that are either just specific to Africans or very different frequency? And boy, do we find a lot. And most of these have not been well characterized. And then speaking of what you said about applying this to psychiatric disorders, one of the papers I had dug up, I don't remember if it was noted in the list that Eric sent, but it was an American Journal of Human Genetics. It was Bogdan Pasaniuk's lab, and it was colleagues exploring depression treatment response by using polygenic risk scoring. And so they were looking at people across different ancestries, and there's been, I think, something that shows that people have major depressive disorder, often are the serotonin, uptake, what is it called again? Drugs used to treat depression. Sometimes they aren't
like
as
SSRIs.
effective. SSRIs, thank you. And so they did a really cool job trying to come up with polygenic risk scores for predicting this across different ancestries. So that's something that people might want to check out as well.
Yeah, we'll link to that as well. Yeah, I think the only other paper I personally wanted to highlight, it's an amazing list, so everyone should look through all of it. It's just because we've talked about this topic a lot on the show over the years, is the paper about exome sequencing and critical illness being useful in adults and not just kids. Because, we've had Dr. Stephen Kingsmore on the show, and he's talked about a lot of, looking at adapting newborn screening to sequencing and different like projects, sequencing babies in the NICU that are critically ill. You and Ashley might still hold the record for the fastest genome. No,
the Broad just broke it 2 weeks ago.
2 weeks ago. See, this is why Eric has to be on, because he knows all the research.
No, the Broad, it was all of the Broad just broke it 2 weeks ago. They have it under 4 hours with interpretation.
Wow. I think you and Ashley was 4 hours and something, or 5 hours, something like that.
Yeah, no, it was all over.
Yeah,
but so Kira, I love that you picked that paper, because if you were going to give us a jump ball later to name what our
that was going to be your
favorite papers, all great, but one that we hadn't talked about. I want to point out two things about that adult ICU paper. Number one, this is where Sarah's going to want to call the Penn Development Office. Penn is just killing it this year. I mean, think about it.
They really are.
First of all, yes. Penn, first of all, has the president of the American Society of Human Genetics as one of their faculty members, and Sarah. Then we talked about baby KJ, and then the very next paper in the article, in the Year in Review article, is this paper, which came out of the University of Pennsylvania Adult Intensive Care Unit. The bottom line is they took sort of a slice of individuals from 18 to 40 something, and they excluded those that were there because of poisoning or accidents or no. And they just said, how many of them have rare genetic diseases? By sequencing the genome, and a quarter of them did. And that's the reason, amazing. And Kira, you accurately sort of stitched it back, but we don't have to go back that far. And it's very meaningful to me because it really was one of the very first clinical programs that I put into place after I became an HGR director, was this exploratory newborn sequencing. Most of the grants we gave out were for healthy newborns, but then we gave this grant to Stephen Kingsmore when he was still at UMKC, University of Missouri.
Didn't even know that connection. Wow.
That's where he did it. And wow, that's just opened up the field. So now there's all this excitement about rapid sequencing of children in, level 4 neonatal intensive care units. Then it spills over. The British and others start looking in pediatric intensive care units, then pediatric cardiac intensive care units. It was only a matter of time. People were going to look in adult, and they did. Not surprising. Why is it some reasonably healthy people get ill and they find themselves in an emergency in an ICU. And it's because some of them are going to have undiagnosed conditions that are genetic in origin. So I think it's really a reflection of how many areas of medicine would benefit from a pre-existing genome sequence. And it's going to elevate conversations about universal sequencing of all newborns. I think the other thing to point out, while we suddenly have found ourselves leaning into this whole idea of sequencing all newborns, three things I'd point out about 2025 as the year comes to a close. Number one, the UK is getting quite advanced in their pilot study of, I think, 100,000 healthy newborns. They've made the declaration that within a decade, they think they're going to see all newborns in the UK. I mean, the UK has declared that, and that was big news earlier in the year. Second big news story was looked on in Florida. There's something called the Florida Genetic Sunshine Act. I may get those words mixed up, but they basically, at the state level, are elevating the priority for rapid sequencing of acutely ill newborns and exploration of sequencing of healthy newborns. And that's Florida. It's not the most progressive state in the world. That's Florida. Yeah. And then a couple of weeks ago, in something that I was involved in giving some advice before I left NIH, the NIH, out of the Innovation Fund of the NIH Common Fund, has a program called Beacons, which is going to be a multi-investigator group, mostly led by Robert Green. It's going to involve state labs. It's going to sequence, I forgot, some number of tens of thousands of healthy newborns, but get it integrated with the already existing infrastructure of state labs doing newborn screening. All of this trajectory points us towards a better and better research agenda that I believe will lead to much more serious consideration of routine newborn sequencing of genomes of healthy individuals. And I think we'll see it in other countries first, and I think the US will follow. And just imagine in so many different scenarios, having all that information available in our electronic health records. So I think I've seen this big ship start to turn a little bit in 2025 when it comes to the idea of universal genome sequencing of newborns.
And I was just going to say there were several papers, actually, American Journal of Human Genetics, ASHG Advances, that talked about, and I don't know if this is different from what you're talking about, the Seek First initiative. That's maybe something
different? These are all interrelated.
They're related, right? And showing that the other thing that happens when you do this more widespread sequencing is helping to eliminate some of these inequities, because one of the things that one of these papers showed is that there tends to be, for a number of reasons, a bias that people of color, for example, were not getting offered the opportunity to get the genome sequencing. But if it became routine and like everybody's getting this done, that would help eliminate some of that bias as well.
Especially like that newborn aspect. If we're just like sequencing newborns in especially New York City, when I think about the United States, like that's probably the most diverse city. It might even be the most diverse city on the planet. I'm not sure. And so just being able to look at that and have so much of that data be representing people from all over the world in terms of their ancestral origin, like that is so, so useful. Before I ask for predictions of 2026, is there any other call outs or anything else we wanted to highlight for 2025? Because I'm impressed how much we've been able to cover in an hour.
Say the two letters that have it. Well, I don't think these two letters really came up, if I remember
Are you
right.
going to say AI?
AI, right? I mean, because I actually think it's relevant to Sarah's recent meeting. I mean, there was no way to escape recognition that permeating throughout the American Society of Human Genetics meeting are little, including on some plenary talks and big talks and little talks and posters, AI, it's just like in our personal lives, AI is everywhere, but AI is starting to become mainstream in genetics and genomics. And so I would say that was the other thing we didn't really emphasize, but clearly is a mark of 2025.
Yeah, I think especially with variant interpretation, that's where I feel like I've heard about it the most, and just being a very like straightforward, this is how it's very helpful of, I've explained this on the show before, that like if you have this list of 300 variants that possibly could be causing the phenotype, if AI can put the actual causative variant at number 3 instead of 300 and we get to that first, like that's so helpful. So like using AI as a tool, but I do worry about that whole aspect of like how much is it replacing aspects that humans used to do. I want to have it be a tool. I don't want it replacing right now, like especially with like clinical testing once we get there and using it.
So there actually was a speaker at ASHG, Matthew Might, who talked about how long it took him to find answers for his son's condition. And then when he turned to AI, he was able to identify the mutation and potential therapies. But I also, and this is part of my, I don't know if we're moving in the direction of the future, but that is definitely going to be part of the future. But it also worries me a bit. I think there are, and not in addition to your question about is it going to replace everybody, there are ethical issues. And there's also the fact that we have to remember, what is AI doing? Okay, it's looking at patterns in data, correct? And then it's a bit of a black box. And many things, it's not telling you what is causing that pattern, right? You still have to figure out what is causing that pattern. And it could be very misleading and it could be very biased, depending on what the data that is put in there. So I truly think that is going to be a major push in 2026. But we also have to have people who are also looking at this from a careful perspective, how to interpret it, how it could be misleading, what the ethical implications are, and so on.
And knowing what data we're supplying. So making
Right.
sure that going off of like echoing more of what you've said, Sarah, of let's make sure that that genomic data that maybe it's searching through to find some answers, to curate some answers or writing, that it's looking at a database that is diverse for what we're focused on, which right now is a point of contention. We're not really there yet. So it does, just like we talked about with polygenic risk scores, like same concept here where it could exacerbate disparities that we already have. And I think everyone's experienced that to a certain extent. Like I was making a presentation for something coming up. And so I was like, well, I want it to generate images for me of just little cartoon of, you know, a genetic counselor and a patient and whatever. And everything it was generating was white people. And I called it out on it. And I was like, I would like you to have a more ethnically diverse depiction. And it said, oh, thank you. But I had to ask it.
Oh yeah, I
So
had the same exact experience doing the same exact thing for some slides I was making. It's like everybody, and yeah, you have to just like the rest of society, you gotta push them a little.
yeah, you really have to ask. I feel like I've gotten really way better in the last 2 years at how I direct AI to come up with things. And usually for me, it's idea generation or aspects for presentations. It's more just, you know, tailoring something, and it's not so much like give me a finished product at all. But yeah, I think we'll see that. And I would predict that one of the papers in the year in review next year will be related to AI, is going to be my prediction, because I feel like it's a pretty safe prediction, so that next year I can be like, I was right. Any other predictions for 2026?
Yeah, I'm gonna give a very realistic assessment. I don't mean this to sound negative, but I think it's really hard to predict what the next year is going to be because I don't know if we're going to still be dealing with, lots of crises and constantly having to put bandages and band-aids and how much of it is going to be, all right, the free fall is over. Now we're rebuilding and we're going to start putting things back to normal. If Sarah Tishkoff has to sit there and spend ridiculous amounts of her time worrying about grants that are getting canceled or grants that have to be rewritten or sanitizing language in grants around diversity and DEI, as opposed to thinking great intellectual thoughts and writing great stellar papers, that's the difference of what the next year will bring, is that these things just suck the oxygen out of the room for these incredibly talented people. And that dictates, and so it has a consequence. And so are we over all this, ridiculous destructive behavior and we could get back to normalcy and just focus on science? And so I think until we know that, it's really hard to say what the next year is going to bring and what's going to feel like it when we all gather at ASHG the ASHG meeting in Montreal next year. And
Yeah,
it's hard to predict. It really is.
Yeah, and that is so true. That is so true. We don't know from, there was an executive order that decisions are now going to be based on political appointees and possibly not based impact score may not have such an impact anymore. So and we don't know who the directors are going to be at the Institute. So there's a lot of unknown, but I do feel like I still can't help it, Eric. I have to remain hopeful. I have to remain that, like, we have to try to stay positive. And I think, one of the other themes of the ASHG was just we need to be there for each other. And I think, Eric, you mentioned that too. It really caused this bonding. We're in this together. We have to support each other. We are going to get through this. It might be a challenging time, but we're going to do it. And we're in it together, and we're stronger together.
That's why a 1 year prediction's harder.
Yeah.
2 to 4 year prediction,
Yes.
all of a sudden find easier.
Well, my job as moderator here is to come up with the hard questions to really push it, just really, really make you uncomfortable, right? Well, thank you, Eric and Sarah, for coming on. This has just been, I look forward to this every year of being able to reflect, look forward, and also decompress a little bit of like what was going on and just being able to vent a little bit. So I really appreciate it. Sarah, for you joining your first time on DNA Today. Eric, I think it's number 8 or 9. So you're just locked in. Unfortunately, you're going to be on a few times a year, every year for the foreseeable future. But yes.
Cheap date, so that makes it easy.
Yes, you are a cheap date. And Eric always he's the one that brings on the current president of ASHG because Eric knows everyone.
And thank you for inviting me. It's really been a pleasure.
Yeah.
Keep up your great work.
Exactly.
Hold your work in such high regard. You really help also a very important part of elevating our field, getting things communicated, and people really do benefit from your efforts. So thank you.
Well, thank you so much.
And to a good 2026 for all of us. It can't be any worse than 2025 at present.
Exactly. And we'll see everybody in Montreal in 2026 for ASHG.
All right.
Absolutely.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.